A warm welcome and thank you all for joining us today for our FSR Grant Webinar entitled Accelerating Sarcoidosis Research Together. Before we begin today, please note there will be a Q&A following today's presentation. You're more than welcome to use the Q&A box, which is located at the bottom of your screen on Zoom, to submit your questions throughout the presentation, and then afterwards our grantees will answer those questions about their work. So just to start us off today, I wanted to share a little bit about FSR for everyone on the call today who's joining us, some of whom may be familiar with our team, but also some who may be new and joining us for the first time. So a little bit about FSR. The Foundation for Sarcoidosis Research was originally founded in 2000, so we're now on our 25th anniversary this year. FSR is the leading international nonprofit organization dedicated to sarcoidosis patients and research. The mission of the foundation is to find a cure for sarcoidosis and to improve care for patients who are living with sarcoidosis. FSR is driven by a cooperative, communicative, and cyclical relationship between our patients and research. So what does our FSR community look like? Well, our community consists of over 70,000 strong from all 50 US states and 80 countries. Our community includes patients, caregivers, family and friends, clinicians and healthcare providers, clinics and researchers, donors, fundraisers, and also volunteers, and also, of course, our staff and our committees. So what do we do here at FSR? Well, there's a few different areas that we specialize in. The first is helping to accelerate research. And FSR does this through collaboration internationally and through our funding for grants, such as our fellowship grant, pilot grant, and other large grant programs as well. FSR also seeks to educate through webinars, town halls, podcasts, and also infographics using information from our experts. FSR also focuses on clinician engagement through delivering educational, networking, and resources to help improve quality of care for sarcoidosis patients all across the world. FSR, of course, seeks to support our patients through patient advocacy programs, patient navigator programs, and also wellness programming. And finally, FSR looks to advocate for increased grant opportunities through NIH and pharmaceutical partners, access to care, and reduction of known barriers in sarcoidosis. And next, we wanted to share about some of the exciting events that have been going on this month and some that are coming up as well. Recently, there was a great webinar last night with Dr. Logan Harbour and Dr. Sumit Sharma from Cleveland Clinic. They talked about other treatments beyond just prednisone, as well as the detection and management of ocular sarcoidosis. This webinar was recorded, and so we'll go ahead and put a link to that in the chat if you're interested in viewing this webinar. We also have some exciting events coming up as well. For example, we have a physical therapy Q&A session, which will be coming up on April 22nd. It'll be at 5:30 Central with our physical therapists, Marian Patesnick and Claire Child. This virtual event is open to everyone of all abilities and also fitness levels. And next, we also wanted to share that if you have pressing questions about sarcoidosis, next week's a great opportunity. FSR is hosting a week-long online educational opportunity for patients to have those questions answered by a leading sarcoidosis expert in the field, Dr. Raj Dasgupta at the University of California, Riverside. This series will take place on Inspire, which is an online vital health community where millions of patients and caregivers can share information and provide support in a private, anonymous environment. And April is, of course, Sarcoidosis Awareness Month. We're now right in the middle of the month, and it's been a fantastic month already. And we wanted to encourage all the different ways that you can get involved in Sarcoidosis Awareness Month. For example, we encourage you to attend a local walk or event that's coming up and possibly volunteer at a local event. We encourage you to light up in purple by illuminating landmarks and homes in purple to help show your support. We also encourage you to take part in the Post in Purple Challenge, where you can wear purple for sarcoidosis, share this on social media, and help us in spreading the word. We also encourage you to visit our Awareness Month webpage, which we'll link in the chat, where you can find different events, resources available to you, and also ways that you can engage. We also have educational webinars, just like today, and our podcast, which we encourage you to check out, the Sarc Fighter podcast, where you can hear from experts and hear about real stories as well. And of course, we encourage you to follow and share your own pictures on social media. Let us know. We have daily posts going out and many different resources to help us in amplifying the message about sarcoidosis. Together, we can make a difference. And finally, we wanted to share with everyone about our upcoming virtual patient summit. This will be June 28th through the 29th this year, and it's entitled United for Progress, A New Chapter in Sarcoidosis. Our patient summit is interactive, it's educational, and it's a networking event for everybody. It'll feature world-renowned expert speakers on sarcoidosis, many opportunities to interact with other patients as well. and we'll cover a wide variety of topics, including disease manifestation, research, and also symptom management. Excellent. So now I'm thrilled to introduce Dr. Louise Perkins, FSR board member, immediate past board president, and chief science officer emerita of the Melanoma Research Alliance. Louise is a healthcare research executive with 3 decades of nonprofit and pharma industry experience. Her passion for research and advocacy programming drives her excellent leadership here at FSR. I'll now pass the presentation to Louise for opening remarks.
Thanks for that kind introduction, Tim, and the terrific overview of FSR. I want to wish everyone on the call a happy Sarcoidosis Awareness Month. As the spouse of someone living with sarcoidosis, I know firsthand that SARC presents significant challenges, but no one should have to face those challenges alone. This year's Awareness Month theme, Facing Sarcoidosis Together, highlights the power of community support and advocacy in navigating this complex disease. Before I introduce tonight's speakers, let me just take a couple of moments to put today's presentation in context. As Tim said, this is FSR's 25th year, and during that time, we've provided over $7 million in research funding to support nearly 50 projects. As a result, over $10 million in follow-on funding has come from the NIH and other large funders. This sort of follow-on funding is important not only for the researchers to continue to follow up on their specific questions, but it also ensures that they stay focused on sarcoidosis. And as an aside, you may or may not know that all of FSR's funding comes from private donations and corporate support. We don't receive any government grants. So if you want to support us or get involved in any way, please check us out at stopsarcoidosis.org. And one way you could get involved could be through participating in our patient registry, which we will be relaunching within the next month. Please consider joining the nearly 7,000 participants from all over the world to provide valuable data about your journey with sarcoidosis to researchers. Now, coming back to tonight's presentation, we're pleased to hear from Dr. Alicia Gerke from the University of Iowa, who was awarded a 2022 pilot grant for her research on how the airway reacts to home allergens in mild and severe sarcoidosis. We're also thrilled to welcome Dr. Emma Thornell, Dr. Gerke's research partner on the project. I'm excited to introduce this webinar for 2 reasons. First, these researchers have enrolled over 60 patients with mild and severe sarcoidosis and used nasal swabs to collect study samples from these individuals. It could be hard to kind of put that in context, but that's really a noteworthy achievement. And the second reason is a bit more personal. I was chair of FSR's governing board when this grant was awarded in 2022. And the way FSR's grant process works is that research proposals are first reviewed by our scientific advisory board, who provides scores and comments. Then the governing board reviews and approves the top-ranked proposals that are recommended by the scientific advisory board. We decided to give out 2 grants in 2022, one to Dr. Amanda Piquet at the University of Colorado, and one, of course, to Dr. Gerke, whom we're here to learn from tonight. So without further ado, I'd like to hand it over to Dr. Gerke and Dr. Thornell.
Thank you, Dr. Perkins. I'm really excited to be here today. I think now more than ever, it's really important for the partnership between patients and physicians and researchers all over to come together and learn from each other. And I will give you a lot of information about what we're doing, but I can tell you with the questions and the answer session, I will likely learn a lot of things from the audience too. And so I'm excited that you're here. I'm heartened that there's so much interest in what we're working on. And I hope to move it forward and continue to figure out how we can better care for our patients. And at some point, really to try to either prevent or cure this disease. But I'd also just like to very much thank the Foundation for Sarcoidosis Research for funding this. This is a pilot study. So what we're presenting today, a pilot study is a beginning work. So it's a grant that sort of gets us off the ground with an idea. and takes it into the bigger realm. And so what we're presenting today is it's data and results and sort of where we're going with this. It's not definitive necessarily, but it has started the cascade of research funding for us. And we're greatly appreciative of the foundation for helping us do that. And it's a great partnership between all of us to connect. So I'm looking forward to hearing any questions you might have at the end of this. And I'd like to point out my partner is Dr. Stapleton and married name Thornell, so that's why it's different. But she has done the grand brunt of this work, and so it was true partnership between environmental health science, me in the clinic, and then the basic sciences over in the lab. So with that, I'm going to share my screen and start our little presentation. All right. Can everyone see the slide?
Yes, we can.
Okay, awesome. So we're going to talk about how the airway reacts to home allergens and mild and severe sarcoidosis. And so we know there are likely a few different aspects about people and the environment and the genetics and how they interact to cause or lead to sarcoidosis. And so this isn't a slam dunk easy answer. We know there are a lot of pieces involved in why sarcoidosis happens. We don't have any conflicts of interest regarding the data presented. So sarcoidosis is a disease by definition of exclusion, and it's because there are many other types of diseases and exposures in the environment that cause something that looks almost exactly like sarcoidosis. And when we find out what that is, what that environmental exposure is, or what that infection is, we don't call it sarco, we call it something different. And so you'll see that for infections. If you have tuberculosis, or in the Midwest, as we have histoplasmosis, which is a fungus. In the Southwest, they have coccidiomycosis, valley fever, and all of those present with a granulomatous reaction in the lung and other parts of your body that look exactly like sarcoid. So we always make sure someone's not infected. You can be exposed to certain drugs. You can get reactions to medicines, and a lot of the ones we use for cancer therapies now cause granulomatous reactions in the body. Metals can do this. So if your doctor keeps asking you if you are exposed to dental ceramics or you work in an ammunitions factory, beryllium can cause something just like sarcoidosis. And then we call it berylliosis when we figure out it's beryllium. And of course, different systemic diseases, different autoimmune diseases can cause a sarcoid-like reaction. So if you have a cancer, like a breast cancer, a colon cancer, or if you have a lymphoma, you can actually present with granulomatous lymph nodes everywhere, and it looks just like sarcoid. But we then call it a sarcoid-like reaction because it's more that your body is fighting this cancer. And so technically, we just kind of separate that a little bit. Well, we don't find any good reason for why this is occurring. We call it sarcoidosis. And that is when we biopsy the nodes, we find this perfect circle of inflammation. These are all inflammatory cells, all focused in one little perfect circle. And then we think, okay, this is likely sarcoid. We can't find any other reason for it. And the pathogenesis, everything that we've studied thus far has implicated that there is an environmental exposure. So that is one of the reasons where Dr. Stapleton and I have sort of trying to figure out, is it, what's the exposure causing it? Or is the exposure sort of not letting something happen in the body? Is it modifying some inflammatory response that's going on? So kind of two different questions. Before, many of us have always tried to figure out what's causing it, but we also entertain the idea that whatever's in your environment might be modifying it, making it worse, or impairing its ability to resolve. And so what happens in sarcoidosis is there's some sort of exposure, and we call this an antigen. Antigen just means it's something in the world. It could be a bug, it could be a metal, it could be a piece of dust that's causing an inflammatory reaction. So your body finds this piece, it presents it to our immune system, and this is called a T cell receptor here. And the T cell receptor is presented this bug or whatever it may be, bug parts, And then it activates the systemic cascade of inflammation. And so there's a very typical inflammatory response. We call it in the lab a Th1 response. And that's a response that's marked against something in the environment where you're fighting something. And this immune response then becomes a big cascade, and it can go away. And so for about 60 to 70% of people, it just resolves. There's something about our immune system that finally says, okay, we need to get this in check. It regulates the immune system, and that's why we call them Treg cells. These are different inflammatory cells that say, okay, it's time to stop now. And then in some people, it doesn't stop. And we don't know why that is, but in a 30 to 40% of people, it perpetuates, and it can be chronic disease, and it can be waxing and waning disease, or it can turn into scar, which is what we are trying to prevent, because it's that scar that you can't get back. And we know there's likely a genetic etiology, and that just means there are certain indications when over studies across the decades that say that's likely somewhat genetically mediated. So incidence and prevalence vary by ethnic group. So, and there's increased risk in families. We call that familial sarcoidosis. About 10% of cases, you'll see it kind of grouped in different families. We've also identified different genetic mutations that are associated with the onset of sarcoid. We've identified other ones that are associated with prognosis, and then certain genetic mutations can be pinpointed to whether you have neuro involvement or just pulmonary, and so we think there might be genetic issue there. We also know that, or we found that a lot of the genetic issues that we find in people are really honed in on this process, or this genetic linkage called the major histocompatibility complex. And this is how we present things to our immune system. So you have little cells around your body mopping up bad things that occur, or anything that you're inhaling, or bugs, and then they're presented to the immune system in sort of, and it's all mediated by this proteins. And this is the strongest genetic linkage we find. We also know that there are different genetically mediated ways that this receptor kind of forms, and that also implicates that there's something that's involved in this presentation and then the reception of an environmental problem. We also know from an epidemiologic standpoint that there's likely some environmental aspect to this, because there was a huge study of over 730 patients called the Access study. It was in the early 2000s, and they looked at environmental risks and did sort of a case control study. They looked at people who had sarcoid versus not. And they found that there was an increased risk of pulmonary sarcoidosis in mold and mildew exposed individuals, and sort of their occupations, associated occupations. We also see that there's been spatial clustering. So we've seen sort of epidemics of sarcoidosis, and one's been reported in a mold damaged building outbreak. We see that it increased risk in US Navy personnel. And we also, most predominantly, this was a huge deal, we're following these people, but the New York City firefighters in 9/11 have a, first of all, baseline increased risk of sarcoidosis, but after 9/11, they have a hugely increased risk of developing sarcoidosis. And that risk is particularly prominent in people who are working on the debris pile, as opposed to ones that were just exposed to the dust cloud. So it implies there's something in this debris pile that might be perpetuating the sarcoidosis. We also know there's some seasonality of sarcoidosis. It tends to spring up in the spring and late winter. And so that always makes us think there might be something in the environment. We also know that if we take cells from patients with sarcoidosis, we take your blood cells, or we take your cells from your lung, and then we put them out into the lab and then subject them to different proteins and different bugs, we see this huge inflammatory response that we don't see in normal cells. And so we know that these cells are primed to react to things in the environment in a way that's overwhelming compared to people who don't have sarcoidosis, which also makes us think that there is something in the environment that's doing this. So overall, we are looking at the different causes of sarcoidosis, but also the different factors that that modify sarcoidosis, that what does make people perpetuate in a chronic way rather than just spontaneously resolve like 60 or 70% of other people. So we're trying to figure out what that distinction is and why that happens in certain people. And it may be different for different groups of people with sarcoidosis. And so that's what we're working on. So we are working on exposures. We know there's a genetic component, and we know that there's different features of the immune system that make people progress. And all of that leads to chronic and progressive sarcoidosis. All right, now I'm going to pass it off to Dr. Stapleton to sort of dig deeper into why we were started to look at molds and bioaerosols, as we call them, and what we found.
Hi, everyone. Can you see and hear me?
Yes, I can, Emma.
Excellent. Alicia gave a really great intro for more of the disease process and background on how that works. I'm going to speak a little bit to the environmental side of things. My background is in air quality, so this is something I've been interested in for a long time, and sarcoidosis has that connection to potentially to what you're breathing in. So we collaborated. So beta-glucan is the cell walls of fungal material, so mold and fungi. And there are these sugar polymers that are kind of abundant everywhere. And the reason we focused on them, in addition to the epidemiological data that Alicia provided, is that there's been some association with sarcoidosis in this material. So a group found once that the lymphatic aspirate from patients with sarcoidosis contains beta-glucan, and that as these concentrations increased in patients, there was more parenchymal involvement of granuloma formation. There are additionally some studies showing that antibodies, so your body's response to Candida and Saccharomyces, so different yeast species, were greater in the BAL fluid of patients with sarcoidosis and in serum than in controls. Another group showed that this fungal specific kind of T cell response, so immune cell response to Aspergillus nigilans, was unique in sarcoidosis in the lungs of patients than controls. And then lastly, this is a kind of arcane method, but there used to be this agent called KFIM that is ground up material from a patient with sarcoidosis that would be kind of injected into a potential patient. And if they responded to it, they were determined or deemed to have sarcoidosis. And some researchers tested this material and found that there was actually high beta-glucan levels. So it's present in fungi, it's abundant, and it doesn't tell you what fungus it is. It's just kind of a biomarker for fungi. I'm having an issue. Oh, there we go. So our study design is that we recruited patients and we gave them this EDC on the right. I'm not sure if you can see my pointer, but it's basically a Swiffer wipe. It's a very simple sampler, and you set that out at breathing height, and through static electricity, particles land on it, and you can test each side for different outcomes. So you can look at fungal exposure and you can look at bacterial exposure, endotoxin. We also asked participants to vacuum up their rugs, and this little attachment has a 40 micron sieve in there, and it collects dust that we can later test on nasal swabs and cells from patients. And this was really supported and helped with Nirvana Metwali. She's a researcher in Dr. Thorn's lab, and she analyzed all of these Swiffer samplers for beta-glucan and endotoxin. We also designed a survey that participants filled out, and that was to try to determine what exposure sources were and how people were kind of living, to see if any of those factors influenced things. And then we compared these findings to biobanked serum from patients and their lung function results, and then whether or not they had pulmonary fibrosis. So Dr. Gerke looked at their clinical CT scans and radiological reports for that part. So here's a schematic. So we had 25 controls in our study. And then of patients with pulmonary fibrosis, 19 had, with pulmonary sarcoidosis, 19 had fibrosis. And then we got blood, we had lung function, and we had CT data. And then we had these home measurements on the right-hand side. So we had specific research questions, and the first one that we hypothesized was that this exposure to what we're calling bioaerosol, basically organic material in the home, so bacterial and fungal cell wall material, is greater in patients with pulmonary sarcoidosis than controls. So these are our findings. You can see that in our cohort, basically the sex and race were not different between patients and controls. Unfortunately, we weren't quite able to match the age. Our control subjects were slightly younger. But then we looked at the bioaerosol concentration. So again, beta-glucan is fungal exposure, endotoxin is bacterial, and both were significantly greater in patients than controls. Our next hypothesis was that bioaerosol is associated with worse lung disease in patients with sarcoidosis. So the first thing we looked at so right here on the x-axis, you have this log transformed concentration of fungal exposure. So fungal exposure on the x-axis. On the y-axis, you have different lung function outcomes. So we did forced vital capacity, FEV1, FEV1 over FVC, and DLCO, all before bronchodilator and the percent predicted, except the ratio FEV1 over FVC. So you can see here that as lung function as beta-glucan, so fungal exposure increases, you see a trend of declining lung function in both FVC and FEV1, but there is no significant finding. So it looks like perhaps there's something there, but it's a little bit unclear still. The endotoxin, the bacterial exposure, I'm not going to, for the sake of time, show those data, but they were just giant circles. There was no association with bacterial exposure. So fungal beta-glucan exposure tends to associate with worse lung disease in patients. So fungus is everywhere. It's found in soil, it's on plants. This plant here on the right with a dog peeping out, that's a plant of mine that had some aspergillus, I believe. And you can see it's on soil. So we were curious, is there anything going on with plants? And I came to this question with my PhD work, actually showed that vegetation and plants are really good for improving air quality, especially for small particulate matter such as PM2.5. So that's combustion particulate matter. Well, they're good for that, but they might not be good for bioaerosol, so organic material. So we asked if plant ownership might relate to fungal exposure and lung function declines. So what we did here, the x-axis looks a little confusing, but basically people either had 0, 1, all the way up to like 50 or 100 plants in their homes. So in order to make this distribution normal, we log transformed the plant number. We added 0.1 so that you can't start at 0. And we compared that to lung function. So we found that FVC and FEV1 were actually associated, their decline was associated with the number of plants that patients owned. There are trends for the others, but again, FVC and FEV1 give us our biggest hits. So these were intriguing results. They're not necessarily causal. We weren't able to quantify the size, the state of the plants, the type of plant. But it's a kind of an intriguing lead that perhaps increased plant ownership might correlate with decreased pulmonary function. So, it's like the old adage, it's not easy being green. They might be good for combustion aerosol exposure, which is really bad for your lungs, but they might not be great if you're really hypersensitive to organic material and an inflammatory responder. So as Dr. Gerke mentioned, with severe disease, sometimes you get the scar tissue, this fibrosis, where these cells kind of lay down collagen and create this stiffness in the alveolar space. And so we wondered, might this exposure to bioaerosol differentially affect disease course in patients with this more severe manifestation as opposed to those without fibrosis? So we, again, looked at this exposure data, the home exposure to fungal antigens. And so the gray line here denotes patients who don't have fibrosis, whereas the black line denotes patients with fibrosis. And you can tell here that FEC significantly declines in correlation with increased exposure to fungus, and only in patients with fibrosis. And that's FVC. The other ones were not significant. You get a slight trend for the non-fibrotic patients with declines in FEV1 over FVC, but so a little more obstruction. But yeah, so we were really intrigued by this FVC decline with increased exposure only in patients with fibrosis. We next asked whether your blood biomarkers and cytokines correlated with fungal exposure. And unfortunately, our sample size is a little smaller here. So we had, for patients without pulmonary fibrosis, we had 22, and those with fibrosis, we had 14. And we were able to assay approximately 80 cytokines, and this was with help from Eve Technologies in Canada. And because these data are inherently, you get a lot of values and you want to make sure you're not falsely discovering something. So we removed any outliers, and then we also made sure that our exposure data were log transformed. So patients without fibrosis, and just to clarify, the sample size will be smaller because we removed outliers. Patients without fibrosis had a lot of different inflammatory markers correlated with their exposure to beta-glucan. So you can see all of them here, and I kind of just spelled out what each one, the bold text indicates they were significantly correlated. But overall, it's kind of like a really broad picture. We have, so your T cell response can be to things like parasites, and that's different from, viral infection to bacterial infection to allergies. All of these, the T cells are really specialized, and they can kind of respond to different stimuli uniquely. And so, as Alicia, Dr. Gerke mentioned, Th1 response is often associated with sarcoidosis. So we have that. We also see Th2 response, which is kind of more allergies, asthma, Th17. So a lot of varied response here in association with residential fungal exposure. However, in patients without fibrosis, only 2 inflammatory markers were correlated with exposure. So there's eotaxin 3, which is this powerful recruiting tool for eosinophils, which are a very classic allergic response inflammatory marker. And then MCSF, which is thought to regulate tissue macrophage and monocytes. So they're kind of different responses, but a much more blunted immune response in those with fibrosis. So in this small sample, we saw far fewer inflammatory molecules correlated with fungal exposure in patients with fibrosis. And we wondered if this might be suggestive of T cell exhaustion. So this slide is Alicia, Dr. Gerke's, and I'll do my best to explain it, but basically there's a kind of a debate in the sarcoidosis world whether the immune response is a little too active or if it might be underactive to exposures. And so in this fibrotic group, it seems perhaps there is too little response. And that's been supported by a variety of researchers who say that basically fibrosis is kind of not an inflammatory disease process. So the evidence for this kind of diminished immune response is that natural killer cells are impaired, T regulatory cells can't suppress this really powerful inflammatory marker, TNF alpha. There's increased apoptosis. So basically your T cells might get exhausted and not respond to the receptor anymore. So next we asked, what might be going on? How are these exposures happening aside from plants? So we kind of mined the survey data that we collected with some help from our clinical and translational center here. And we found two things kind of stood out. There's a few trends, but cat ownership led to increased exposure to fungi, as did cooking. Now, cooking might sound like a strange one, but when you cook, you generate a lot of particles, and these particles can independently create problems, or they can bind to other things in your home. So we observed increased exposure with cooking and cat ownership. And now just brief conclusions for this pilot work is that bioaerosol exposures are higher in homes of patients than in controls. There was a correlation with lung function decline and residential fungal exposure in patients with fibrosis. We didn't observe any correlation with bacterial endotoxin, so that was really interesting. So it kind of gave us a little more validation that our results we were kind of getting onto something, hopefully. And so this, as I mentioned, the T cell response, you can have a variety of types of response to different antigens. And we found that all of a variety of different responses correlated with exposure. So it wasn't a really clear picture there. But in general, there were fewer inflammatory molecules correlated with exposure in patients with fibrosis. So we asked if perhaps the T cells are tired. And that's beyond the scope of this work, but that's our hypothesis moving forward. And we'd like to, in the future, work with larger and more diverse cohorts. We have a grant application under review that would include a group at Johns Hopkins, and then establish mechanisms of disease modification by indoor beta-glucan. And perhaps, sarcoidosis, as you figure out different triggers, they get called different things, as Alicia had mentioned. We're wondering if we could identify a cohort that might be sensitive to beta-glucan. And then eventually look into cleaning the indoor environment and seeing if that improves disease course. And I have to credit Dr. Gerke with this slide. This is hers. And we'd really like to appreciate and acknowledge the participants who were critical to making this work successful. It's currently under publication, and the Environmental Health Science Research Center, Dr. Matt Wally and Peter Thorne. And then in pulmonary, for the initial work, Dr. Cameas was really helpful. And Dr. Puzzulo has been really critical with the nasal cell exposures, which we weren't able to discuss today. But this pilot funding, as I mentioned earlier, is so helpful for us, and it really got us set up for success. And I'd just like to point out that we're kind of learning things as we go, and we're partway up the tall mountain of how sarcoidosis will eventually, hopefully, be cured.
Fantastic. Well, thank you so much, Dr. Gerke and Dr. Thornell, for your excellent presentation today, and just for all the incredible work that you're doing and for sharing this with all of us in our community. So now we're going to go ahead and begin our Q&A portion for the evening with some questions that have been coming in from our audience. So without further ado, we'll go ahead and get us started. So we are getting some great questions in the chat. Just to kind of start us off, Dr. Gerke, what inspired you to pursue this area of sarcoidosis research?
Well, sometimes it ends up being meeting a person who has a curious interest and figuring out how it might apply. And so I met Emma in the lab, and we started discussing sarcoid and the environment, and thought, she has novel ways of checking these things that I had not been aware of. And so it just became a natural, well, sarcoid's environmental. She loves the environment. And so we should study this. So it actually was sort of like bumping into someone at the cafeteria.
Fantastic. Thank you so much. And next question for you, Dr. Thornell. What prompted that specific focus on dust and mold exposures, among other sorts of pathogens out there?
Yeah, sure. So bioaerosol has long been thought to contribute to lung disease, especially like asthma. And so LPS and mold are both common antigens in lung disease, and they're studied in depth by colleagues. And part of my PhD work was in measuring these in farmers. And so, like Dr. Gerke mentioned, sometimes it's just who you meet and when and how it's easy to work together. And so I had worked with Dr. Thorne and Dr. Metwally, who I mentioned earlier, who are really leading experts on bioaerosol exposure. And we had previously observed in a cohort of female cooks in India that this beta glucan exposure was correlated with bronchovascular bundle thickening, which is a CT imaging finding of thickening of your airway. So they were intriguing. And so then we kind of worked with Dr. Gerke and we thought about how could this be involved in sarcoidosis. And so over the years, we got this off the ground, and we started during COVID. So it took a little while to get off the ground, but yeah, it's been really rewarding.
Fantastic. Thank you very much. So next question back to you, Dr. Gerke. What can patients with sarcoidosis do to possibly control those bioaerosol concentrations in their homes? Are there any specific precautions that could be taken to help in mitigating that exposure?
Yeah, probably a good question for Dr. Thornell, but I would say that, this data is preliminary, so we can't say it's exactly causal or if it's associated with something else in the environment or how it's actually affecting the disease. So I'll say that front. So we hope to figure this out. But what I tell my patients now is, common sense modalities. If you have black mold, hanging out in your house, in the bathrooms, in your closets, it's probably a good idea to clean it up. But as far as other mitigating factors, Emma, do you want to comment?
Sure. Yeah. As Dr. Gerke mentioned, it's kind of without actually going in and measuring your home and everything that's going on. It's hard to say. And it's also not, we don't know for certain that this is gonna be bad for you. And there might be just a few people who are really susceptible. So this is pilot work. So we don't want to kind of overstate anything, but general good practice for air quality in your home, there's a variety of things you can do. So you want to make sure that your air filter in your house is really MERV 13 or above. So that just means how tight the filtration is. So it allows fewer particles to get through. It's a little more energy intensive, unfortunately, but it's better for purifying your air. And then, thinking about things that could contribute to your exposure. So I mentioned plants, just making sure they're not moldy, keeping your pets kind of clean, and anything that goes outside can bring it inside. So that organic material then gets inside. Even carpeting can really harbor a lot of materials. So just keeping those really clean. One thing I definitely do, and Dr. Gerke does, is when we cook or our partners cook, we turn the exhaust fan on in the kitchen, especially if it vents outside, because you really want to pull those particles out when you're cooking. That's one of your biggest exposures at home, is actually that combustive process of cooking. If your fan doesn't exhaust outside, you could always invest in a secondary air purifier. I would recommend a HEPA filtration system. The ionizers kind of generate ozone. So yeah, let me know if you have other questions, but that's just kind of what I personally do. It's not necessarily medical advice, but I think, we should all be kind of cognizant of what we're breathing in. And if the air quality is good outside, feel free to open your windows, but that also, if it's super damp, you might introduce water. So something to keep in mind.
Fantastic. Thank you both. Yeah, it's very good practical advice to just kind of keep in mind and be aware of, right, what's in our surroundings. And kind of to that same point, and Dr. Thornell, if you'd like to take this one, for pathogens, maybe outside the home, but maybe in the workplace based on, different professions, and I know it may vary per profession, but there are any general extra precautions that can help to be aware of what exposures might be present there for those patients?
Yeah, it's so workplace specific. I think if you're able, and it depends how great your exposures are. So whatever you're exposed to, what's really going to lead to effects is the dose over time. So if you have this constant exposure, that's like a very specific thing at a high concentration over time, that's something that you would likely want to mitigate. However, most of us in the real world kind of work with a complex mix of exposures. So it's really hard to say. You hope that your workplace has good ventilation. Industrial hygienists really are the experts in occupational exposures. And NIOSH is a real wealth of resource for that. Unfortunately, they just lost their funding, but I guess for us, I always make sure that the air is ventilated. If I'm smelling something, if it's really smelling toxic, I kind of step away for a moment if you're able. But you can look up like NIOSH stats on exposure limits, and I guess that would be what I would recommend.
That's wonderful. Thank you so much, Dr. Thornell. And Dr. Gerke, next question is for you. So you had mentioned that sarcoidosis is believed to be caused by, that combination possibly of environmental exposures and genetics. Should patients consider asking their doctors for a genetic test? Is there things available for them?
So we don't know how to use it in the clinical realm yet. It's given us, again, hints that there might be some genetic, potential modifications of the immune system or more predisposition in one way, but we do not have one genetic test that will tell you, are you going to progress? What kind of drugs should we use? Are you going to get sarcoid? We just don't have that right now. So it's a little bit behind, let's say idiopathic pulmonary fibrosis, where there's some actual, familial true genes that you can test for now. But so sarcoid, we don't have that yet, and we don't understand how to use it in the clinical realm. So this genetic work is all based on very large studies that were done, mostly a lot with patients in this AXIS study. And so they give us hints here and there, but they aren't clinically used at all yet.
Thank you. And next question for both of you. So we've seen a few questions about the next steps in this research. Do you see this kind of study being conducted for other forms of sarcoidosis as well, like cardiac or neurosarcoidosis?
I mean, we do have actually, sometimes when you start to really phenotype these patients, and phenotype means that you describe the different organ manifestations, you describe their different pulmonary manifestations. And occasionally we do have some preliminary data in other realms where we'll see some exposures really be predisposed or really elevated in someone with, let's say, cardiac only sarcoid or just has cardiac sarcoid. And so I think there's great potential as we start to really hone in on finding different groups of patients, because as we know in sarcoid, everyone's different. And so that's what makes it so hard to study, because you can't put people in a box and then, say, is this associated with this box? Is this associated with that box? And so we are starting to try to find those boxes so we can compare. But every now and then we find some hints, and we have, but not mainstream yet. But it definitely has potential as we now pick apart the clinical characteristics.
Absolutely, thank you.
Yeah, and Dr. Gerke really spent a lot of time looking at each and every patient, and we have just Excel spreadsheets of, what they're afflicted by and, how that translates to their clinical care. So she did a really great job with that. So hopefully someday we'll be able to figure it out. But yeah, it's so diverse and there's so many variables to look at in a small sample size. So it makes it difficult.
Yeah, thank you both. It's fantastic. And next question for you, Dr. Gerke. Can prolonged exposure to these particles in the home possibly lead to that condition of fibrosis? Is it recommended possibly for more frequent testing or potentially medical visits to watch for how that disease is progressing if it might be suspected that there's more of that exposure in their home?
Given that this is really preliminary data, I wouldn't make any solid recommendations, but generally speaking, doctors should be following patients with sarcoidosis fairly regularly anyway, with pulmonary function tests and reassessment of symptoms, and looking at different measures of organ involvement. And so, following those common sense sort of rules about cleaning the home environment and breathing in good air, but also being followed regularly by a doctor who knows you and who knows what to measure is very important.
Great, thank you. And another question for you, Dr. Gerke. How would someone be able to sign up in a potential next study that you might be driving and working with Dr. Thornell as well?
Yeah, so we are, we have applied for government funding, which is sort of up in the air right now. And so as soon as we're up and running, we may be adding centers, and hopefully we will get funded. So right now we don't have sort of, unless you're at the University of Iowa, any active recruitment, because we're not allowed to go outside of the University of Iowa. But should we get funded, then I think you'll be hearing a lot more about that. And that's what's important about these collaborations with like FSR, because we can share what we're doing, have patients contact us, and then bring us together.
Wonderful. Thank you. And we'll look forward to hearing the news there as time goes on. And next question here, maybe Dr. Thornell, if you would like to start. Any final comments or takeaways from today before we kind of close out here? If you'd like to begin, Dr. Thornell.
Sorry. So, we're just really grateful to be able to study this devastating disease. And it took a lot of time and effort on behalf of a lot of people to really volunteer to, put these monitors in their home, vacuum their rugs, send it back, follow up with us, inquire about their exposures. And I guess I want to just encourage that kind of eagerness to research and discovery. And hopefully this will lead to some helpful interventions long-term for patients suffering with sarcoidosis. But no, we're just really grateful to have the opportunity to work alongside everyone.
Wonderful. Thank you so much. And Dr. Gerke, if you would like to weigh in as well, any final comments or takeaways from today?
Overall, I think that it's important to really stay up to date in all these studies. There's a lot of neat work being done in sarcoidosis. There's a lot of movement behind it. I think there's a lot more awareness about what sarcoidosis is and what it does to people. And so I think as we go along, we're increasingly asking patients to participate, to comment, to just tell us their thoughts of what we should be studying and what's important. And so I think it's really important to make these collaborations, also just discussing your care, being open, asking questions to your doctors. And I think that improves the care. And we hope overall in the future that what we do will help change this future for patients with sarcoid.
Well, thank you both so much again for sharing about your incredible work today and this very crucial pilot study, as we're still understanding more every day about how sarcoidosis works and how the environment plays a role. So thank you truly so much, both of you, for joining us today. And I'm going to go ahead at this time. I'm going to pass it over to FSR's CEO, Mary McGowan, for closing remarks.
Thank you, Tim. And thank you, everybody, for joining us for tonight's webinar to learn about one of the research projects supported by FSR. It was so exciting, so enlightening to learn about environmental exposures. We have invited all grantees to join us in these kinds of events to translate their work back to the community. So please keep an eye out for more grantee webinars in the future like this. We invite clinicians to join FSR this coming Wednesday for our clinical education and engagement series on bone health and mineral metabolism. This virtual session will take place on April 23rd from 4 to 6:30 p.m. Central Time. The program is open to clinicians only. To learn from experts on current research and best practices for bone health and mineral metabolism in sarcoidosis. And clinicians do receive CME credits if they attend in person for this. So if you're a patient, we would appreciate you sharing this opportunity with clinicians at your institution who may be interested, including orthopedics, rheumatologists, endocrinologists, and others. So we're really looking forward to that on Wednesday, and you can share that the program will be recorded and sent to all the registrants, even if they can't attend live. I'd also like to put in another plug following Tim's introductory remarks highlighting FSR's upcoming FSR Global Sarcoidosis Clinic Summit on June 28th and 29th. This is really more than just a conference. It's a space for patients and caregivers and clinicians and researchers to all come together. And that's where true progress happens. So whether you're living with sarcoidosis, supporting or caring someone living with sarcoidosis, this virtual event is definitely for you. And we're excited. Early bird registration is open until May 15th. And in celebration of FSR's 25th anniversary this year, there is a reduced registration fee to just $30 a person. And we do anticipate over 400 people from around the world attending this event. So please mark your calendars. We hope to see you there. And in closing, I would like to thank Dr. Perkins, Dr. Gerke, Dr. Thornell for sharing with us this evening. And for all of the attendees' thoughtful questions, thank you so much for your engagement this evening. We'll be sending out a post-event survey, and we hope that you'll take just a few moments to complete the survey and to let us know what you thought about this evening's webinar and inform us of other kinds of research updates that you would like to hear in the future. So with that, thank you so much again for joining us, and we hope everybody has a wonderful evening. Thank you.